as an introduction, so if you look outside in nature, in particular now in early spring times, you will see that the world is a largely green place. And this is despite the fact that plants are basically constantly challenged by different types of stresses, including abiotic stress such as heat or drought, and also, which interests me most, biotic stress. So biotic stress imposed on plants by pathogens or potential pathogens that try to infect plants in order to gain nutrients from them and make them sick. And these includes various types of organisms, including bacteria, fungi, oomycetes, viruses, and also larger organisms such as nematodes. And the reason why plants can cope quite well with the majority of these potential pathogens is because they have evolved a sophisticated innate immune system, which allows them to detect these microbial potential pathogens to activate an immune program. And the first line of induced defense of plants against attacking pathogens is also known as pattern-triggered immunity, or short PTI. And this is based on the perception of highly conserved molecular signatures derived from those microbes by plasma membrane or cell surface localized pattern recognition receptors. And this then results in the activation of a downstream immune program, which enables the plant to fend off most potential pathogens. And depicted here on this slide is a hallmark example that I'm also studying a lot in my group, which is the perception of bacterial flagellin, so which is derived from bacteria flagella, via the FLAG22 peptide epitope by the receptor FLS2. So in response to FLAG22 binding to FLS2, a rapid ligand-induced receptor complex is formed, which activates downstream signaling events, such as, for example, the influx of calcium ions into the plant cell, the production of reactive oxygen species, the activation of downstream kinase cascades, which ultimately results in a transcriptional reprogramming of the plant cell, and also in the production of, for example, phytohormones involved in immunity, such as ethylene or also salicylic acid. And this all together confers pattern-triggered immunity to fend off most invading pathogens. But the activation of immunity is not always beneficial for the plant, because it is impossible for the plant to simultaneously grow and defend itself. And this is illustrated by the so-called growth-defense trade-off, and shown here by two examples from the model plant species Arabidopsis thaliana, which I'm also doing most of my work in. So as you can see in the top part, there are Arabidopsis seedlings that are grown in the presence of immunogenic elicitors, such as the aforementioned FLAG22 or another related peptide. And you can see that their growth is strongly compromised in the presence of these peptides compared to control-grown seedlings. And here on the lower side, you see a mutant which produces a constitutively active defense protein. And this plant is very resistant against different pathogens, but obviously it doesn't look very happy. And the growth and development of this plant is strongly compromised. And therefore, there need to be mechanisms put in place in order to regulate this trade-off between plant growth and plant immunity. And this is what interests me in my research most. So I've shown you the left part here on this slide already. So you can see that microbes are perceived by these pattern recognition receptors, like, for example, this FLS2 example. And these proteins belong to the family of receptor kinases. So they contain an extracellular ligand binding domain and an intracellular kinase domain, which is required for the signal transduction down into the cellular compartments. But interestingly, very similar looking proteins, so other receptor kinases, are not involved in defense, at least directly, but they are regulating diverse aspects of plant growth, development, such as also reproduction or nutrient sensing. And they do this by perceiving endogenous signaling molecules, including, for example, the famous prasinosteroid growth hormone, or a plethora of different endogenously produced plant peptides, which then bind to these receptors to activate signaling pathways mainly involved in growth and development. But to increase complexity, there's emerging evidence from the last years that these primarily growth-related receptors, together with their endogenous ligands, also fine-tune or modulate signaling conferred by these pattern recognition receptors during plant innate immunity. And I'm interested to study this signaling crosstalk. And peptides that can fulfill such a crosstalking function are also referred to as phytocytokines, as Tina already said in her introduction. And this is based on their analogy to metazone cytokine peptides that you probably all have heard of as important regulators of immunity. And they are not evolutionarily related at all, but they share several common features. Like, for example, both types of peptides do not only regulate immunity, but also growth. Their biosynthesis or secretion can be modulated after danger perception. They are recognized by specific receptors, and then they can function in an autocrine or paracrine manner, which means that they can affect their own secreting cell or tissues or cells distal to the actual secretion site. But importantly, the mechanistic basis of signaling crosstalk of these phytocytokine pathways with innate immunity remained largely unknown. So upon starting my own group now, I was first interested to identify novel types of these phytocytokines and then use them as a molecular toolbox to study their signaling crosstalk with immunity. And for doing this, I actually started with a fairly simple transcriptomic approach and screened publicly available gene expression data for known growth regulatory plant peptide families and whether there are specific members that show a differential transcriptional regulation after either infection with different pathogens, like for example, Pseudomonas syringae, an important bacterial pathogen, or treatment with immunogenic elicitors. And while doing this, I came across a peptide family which is known as the Golvan peptide family. And as you can see here, this family consists of 11 members in Arabidopsis, and 2 members in particular, Golvan1 and Golvan2, showed a differential transcriptional regulation after infection with these bacterial pathogens. So what are the known roles of these peptides? They've been identified by different groups around a decade ago to be important for several aspects of root growth regulation in Arabidopsis. So they have also, based on these different groups that have identified these peptides, a quite complicated nomenclature and have 3 different names. So not only Golvans, but also RGFs and Cli-like peptides. But I will only refer to them as Golvan peptides for the sake of this talk. And they were initially identified to be important regulators of root stem cell niche maintenance. So they are required to maintain a certain pool of root stem cells, which is really important for root growth and root elongation. And then they were also found to be involved in root gravitropism. So normally the root grows towards the angle of gravity, but if the peptide levels of these Golvans is disturbed, this stimulus is kind of disturbed and they show this wavy growth pattern, where also actually the name Golvan comes from, because Golvan is a Dutch word for wave. And they were also found to be involved in lateral root emergence, so not only primary root growth, but also lateral root growth. But importantly, there was no known role of these peptides in leaf tissue, where we usually do our immune assays with, and no known or described role in the context of plant immunity. That's why we decided to study this peptide family in more detail. So first of all, we were interested now to test whether indeed Golvan1 and Golvan2 may be involved in flagellin or FLAG22 triggered immunity. And we first did this by analyzing overexpression lines, which overexpress both Golvan1 or Golvan2. And then we noticed that indeed these plants have a differential behavior after FLAG22 stimulation, as indicated here in this Ross burst experiment, where we measure the production of reactive oxygen species upon elicitation with FLAG22. And you see the black curve is a wild type here, and all these Golvan1 and Golvan2 overexpression lines respond stronger to elicitation, suggesting that they may play a positive regulatory role in FLAG22 triggered immunity. We then went ahead to confirm these overexpression data by genetic loss of function studies, and we genetically eliminated Golvan1, Golvan2, and other related peptides that are expressed in leaves by CRISPR-Cas9 technology. And we indeed found that this CRISPR Golvan mutant now responds weaker to FLAG22 elicitation, as shown here in this ethylene accumulation experiment compared to the wild type. And they are more susceptible to Pseudomonas syringae infection, as shown here are the quantification of bacterial titers upon inoculation of these plants with Pseudomonas syringae. So engolfin peptides are produced as larger propeptide precursors and require several steps of post-translation modifications for peptide maturation, including proteolytic cleavage of the precursor, as well as tyrosine sulfation and hydroxylation of a conserved proline residue. And so we were interested whether we can actually detect the golvin-2 peptide also in leaf extracts, so in apoplastic wash fluids, which only contain the extracellular space, so what the plant cell actually secretes. And we did this based on a chlorophenol-based peptide purification method, which enriches for small peptides from these apoplastic wash fluids. And afterwards, we omitted any kind of digest and detected or tried to detect these peptides in collaboration with Tina here at BayBioMS using parallel reaction monitoring. And indeed, we were luckily successful and could detect this peptide in wild type plants, so shown here. And more strikingly, the levels were strongly increased in overexpression lines of this peptide precursor gene, really suggesting that the overexpression also translates into more biologically active peptide in the plant apoplast. We also tested whether either FLAG22 stimulation or bacterial infection affects the secretion, but unfortunately this was not the case, and we are still trying to investigate what that means. So we then tested whether this biologically active golvin-2 peptide is also able to stimulate or activate immune responses. And what we noticed is, first of all, that the peptide itself did not trigger any immune responses. So it did not, for example, result in ethylene accumulation. But interestingly, co-treatment of the peptide together with FLAG22 promoted or enhanced FLAG22-induced responses, suggesting that the golvin-2 peptide boosts FLAG22 triggered responses, which also fits to the genetic data we previously observed with the overexpression lines or the loss of function mutants. So the next step for us was then to identify which receptors are involved in golvin-2 perception to regulate immunity. And here we got some hints from the literature because related peptides were shown to be perceived by a family of 5 leucine-rich repeat receptor kinases named RGIs. And so we obtained an RGI quintuple mutant and first noticed that this mutant responds also weaker to flagellin in different bioassays, including this ethylene accumulation experiment. But more importantly, this mutant was insensitive to this golvin-2 effect. So there was no promotion of FLAG22-induced responses upon co-treatment in this RGI quintuple mutant. And consistently with the role in immunity, this quintuple mutant was also more susceptible to bacterial infections, actually to similar levels as the FLS2 receptor mutant or an important FLS2 co-receptor double mutant that is strongly compromised in immunity. So the strongest expressed of these 5 receptors in leaf tissue is named RGI3. And so we tested whether the ectodomain of RGI3 produced from insect cells can directly interact with the golvin-2 peptide. And we did this in collaboration with Yusuf Belkadir at the GMI in Vienna by microscale thermophoresis. And indeed, we could show that RGI3 ectodomain binds to the golvin-2 peptide with a high affinity, so a KD value of around 11 to 12 nanomolar. And this interaction is dependent on this sulfated tyrosine residue, because if this is mutated, the interaction strength is decreased by a factor of roughly 10. So, and then we were interested to elucidate what may be the molecular mechanism. So how do these golvin peptides actually promote PTI? And in previous studies by other groups, they have shown that golvin signaling can promote the abundance of certain proteins, like, for example, the auxin efflux carrier PIN2 in the regulation of root gravitropism. And so we were wondering whether golvin-2 may also promote or regulate the stability or accumulation of important signaling components in innate immunity. And so we tested, because we have native antibodies available, whether Golvan2 treatment or overexpression may affect the abundance of either FLS2 or its co-receptor BAC1. And indeed, we found that the FLS2 levels, as shown here in the immunoplot, are increased in response to Golvan2 treatment, and which is mimicked also by the overexpression of the peptide precursor. But this does not seem to be a general effect, as the levels of the BAC1 co-receptor were unaffected. And interestingly, this happens on a post-transcriptional level, because the transcript abundance of FLS2 is not affected by the Golvan2 peptide treatment. And consistently, the RGI quintuple mutant now showed also a reduced FLS2 accumulation, which can be complemented by expressing RGI3 in the mutant background. So, and as FLS2 and RGI3 are both receptors, we were interested whether they are also able to interact by co-immunoprecipitation experiments. And we found that under untreated or unchallenged conditions, there is no interaction between these two proteins. But in response to either FLAG22 or Golvan2 treatment, we see a ligand-induced complex between FLS2 and this RGI3 receptor, which may underlie the role of these RGIs to regulate subsequent FLS2 stability or turnover. And so the future research directions in this project are to mechanistically understand how FLS2 stabilization is regulated via this Golvan RGI signaling pathway, to understand how Golvan maturation is regulated during immunity. Then we are interested to study more into tissue specificity. So where are the peptides and the receptors expressed, to get an idea whether they function autocrine or paracrine. And then how do they actually integrate cross-related stimuli with innate immunity. And with this, I'm finished and want to thank several people. So first of all, Ralf Hückelhoven, who is the head of our department, who allowed me to establish my own line of research in his department. And then my PhD students, Jakob and Henriette, then my amazing mass spec collaborator, Tina Ludwig, which I'm having still a few other projects ongoing with, and my collaborators here from the University of North Carolina and from the Gregor Mendel Institute, and you for your attention. Thank you very much.
I can see it, right? Okay, good. Yeah, so nice. Tina's very, very thoughtful introduction. So today, so instead of telling you about what my group actually doing today, I'm going to talk about my project when I was a postdoc at Chair of Proteomics with Professor Kuster. And this topic is, so I'm very looking forward to share with you this work. And this is, in this work, we are going to, we actually want to understand the mode of actions of a kinase inhibitor in sarcoma cells by connecting the phospho and the full proteome, baseline proteome, and drug responses. So the main objective of this project is actually to understand the cell mode of actions. And why is that important? Because tumors are often molecularly heterogeneous and show various drug response. And even though we have a lot of cancer drugs, a lot of them actually has a polypharmacologies that result in the different mode of actions. So to understand this, it will pave the way for the future application in the precision medicine by using these drugs. And in the past, people already connecting for example, gene expressions or protein expression with the drug response. So in this study, we're especially interested in the kinase inhibitor because first, phosphorylation is often dysregulated in the cancer. And we want to also add a dimension of the phosphoproteome in this analysis. And by connecting each other and also drug response from the kinase inhibitor, we want to learn more about the mode of actions from those kinase inhibitors. So basically, we ask a question: what can we learn about the mode of actions by connecting the baseline full proteome and phosphoproteome with drug response? So maybe I can have the pointer here. So it's a very busy slide. So to generate data set for this study, we actually use sarcoma cell lines as a models. There are many reasons. First, the genomic profiling of the sarcoma is often not very informative. And also it's because they have a very low mutation rate compared to the other cancer entity. So we chose 17 different sarcoma cell lines from 12 entities and perform the high throughput drug screening. As you can see here, we include 150 cancer drugs and 139 of them are kinase inhibitor. So of course, we also have some chemo drug or the other non-kinase inhibitors. And this would generate roughly 2,550 phenotypic screening, dose-dependent drug response. And meanwhile, for the drug, we also mainly focus on clinical approved drug and the drugs that are the phase 3 clinical trials. So we can say mainly most of them are already being used in clinically. And meanwhile, we also generate the baseline deep full proteome and phosphoproteome by having a different fractionation by the high pH HPLC fractionation. And this data set actually give us roughly 9,000 to 10,000 protein group per cell line, and also 10,000 to 27,000 phosphosite. And here you can see the drug response from the 150 drugs here and across 17 cell lines. And the scale of the color shows you the area under the curve. So which means that if the color is more blue, it's much more potent. And if the color, another way around, if the color is more red, it means that this drug does nothing on the cell line. As you can already see that there are several clusters, so you can pay attention to the right cluster that they are mainly the chemo drug and the CDK inhibitors because they are targeted the common mechanism of the cells. But we also do find some cluster like this one and also this one, they only work on certain cell lines because they are very specific molecularly targeted drugs, and they only target a specific mechanism when the cells have this mechanism existing. So now we can have an idea about the potency. And here's the example of the pazopanib. So pazopanib is one of the multi-tyrosine kinase inhibitor, which has been proved for sarcoma treatment. And you can see that, just take 3 cell line as example. You can see that we have a cell line which does not respond to pazopanib, and we have a cell line which are semi-respond to the pazopanib, and also the cell line which is very sensitive to pazopanib. And these two type of the curves that we can calculate the EC50, and the blue line here, they can roughly give you EC50 100 nanomolar. So we can say that across 70 cell line, we do find many drugs that they do have a very high potency. So next step, we decided to connect the drug response and the proteome data. So how are we going to do here is to calculate the activity landscape of each cell line by combining different things. So we combine kinase abundance from the full proteome data, and we combine the kinase phosphorylations and kinase active loop and kinase substrate phosphorylation. So basically we want to know how active of the kinase, of a certain kinase in this cell line. And as you can see, the heat map here, we were able to calculate 383 kinase activity score in each cell line. And you can already see the activity of each cell lines are very different from the others. And we assume that if a kinase has a very high score in one cell line, let's say more red here, and this cell line should be very sensitive to the kinase inhibitor that target this kinase. And we do find that in some cases, this is true. For example, when we look at the FGFR as a FGFR inhibitor as an example, we have G401, which is a rheumatoid cell line. And when we rank all the kinase from high to low, and we find that PDGFR beta and FGFR2, they have both very high score. And compared to the other fibrosarcoma cell, they have relatively low, below 0 activity score of these two kinases. And if we try to find out what is drug response in our big screening data set, you can see there are 4 FGFR inhibitors has been tested in our data. And the y-axis shows you the AUC. As I mentioned, the lower the AUC, the more potent this drug on this cell line is. And you do see that the G401 in general are very sensitive to the FGFR inhibitors compared to the HT1080. Since, as I mentioned, that we calculate the score of the kinase, but not all the kinase have targeted inhibitors. And we then specially focus on the top 10 druggable kinase. As you can see, the list of the cell line on the SW6A4 is also a fibrosarcoma. And then there is a second highest score is a PI3K. And within our drug screening, we do find that this cell line has a very high sensitivity to PI3K inhibitor, which is even higher, even sometimes more sensitive to PI3K than the chemo drug. And as I mentioned before, our screening mainly focus on the kinase that are clinically approved or at the phase 3. So you can imagine that we did not cover all the druggable kinases. So we then use the example on the KJOS MP and try to find out if there's any drug can target most of the top 10 kinase actively in this cell line. And we find 2 kinase inhibitors at the phase 1 clinical trials, and they do show quite good sensitivity compared to the, in general, 150 other drugs. So we do show that by using this kinase score, we were able to suggest a more effective drug to inhibit the cell line. And the next thing to connect to the proteome and full proteome and the drug response is that we perform the elastic net regressions. And in this analysis, we will see how often a protein, so each dot is a protein of a phosphosite, how often of this protein or phosphosite associate with the diverged drug response. So one example is cobimetinib, which is a MAP2K inhibitor. And y-axis, you can see how much times of this protein or phosphosite were picked when the analysis was performed 100 times. So the higher the frequency means that this protein or phosphosite is highly associated with the drug sensitivity. And then the effect on the x-axis, you can see here, it indicates you whether they are related to resistance or sensitivity of this drug. And then in this case, we find a lot of downstream and upstream protein and phosphosite in the MAPK signaling pathway. And we even identify the MAP3K, which is the upstream of the upstream kinase of the MAP2K. And we also find a MAPK1, which is downstream kinase of the MAP2K. So it's pretty well recapitulated the MAPK signaling by this analysis. And the second example I want to show you is that Vigatinib is an FGFR inhibitor. And we actually find quite a lot normal proteins or phosphosite involved in the FGFR signaling pathway, which has never been mentioned before. So to prove that if these proteins or phosphosite are really regulated by Vigatinib, we perform a time-dependent Vigatinib treatment on G401 cells, which is, as I mentioned, is rupadomic cells, and they are very sensitive to FGFR inhibitors. So in total, we have 11 time points ranging from 5 minutes to 24 hours. And each time point, we even include a DMSO control. So this plot shows you the fold change of a certain phosphosite compared to the time point 0. So after drug treatment, the downregulation of FGFR phosphorylations and also stage 3, it's a known downstream of the FGFR signaling, they are downregulated compared to the DMSO control. And we actually additional observe 2 kinases, ST2 and WNK1. They also show the downregulated after the FGFR2 inhibition. So to summarize, we show that the integration of the drug response and the phosphoproteins, full proteomes, can identify the molecular markers that often explain the drug sensitivity or resistance. Although we can generate quite a lot of hypotheses based on our observations, we also are aware that the correlation is not causality. So if we have a new hypothesis, we need the experimental validations. So in the future, this data set, including, let's say, drug risk screening, elastic net regressions, will serve as a rich resource for public by the interactive use of photo. And also the mode of actions we learn from this study is also one important step for the molecular patient stratifications. Actually, another ongoing project, Topaz, we aim to give an individual treatment recommendation according to the proteome activity in sarcoma patients. And understanding the drug mechanism of actions is definitely one of the key steps to advance the precision medicine. So with this slide, I would like to finish my talk, and I would like to thank the people at the chair of proteomics and bioanalytics. As I mentioned, this project I conducted when I was a postdoc at the chair, and I really appreciate all the people, all the co-author and all the other people have been helping me, and also I had a great time there. And I also I would like to thank Kerstin and Joe at the EMBO Chemical Biology Co-Facility. They performed the drug screening together with me. And I would like to thank for your attention, and I'm happy to answer any questions.
Okay. Hello everyone, and as was just so nicely introduced, I am Biene, and I'm a PhD student in the AG Piegelmeier at the Institute of Virology here at the Technical University of Munich. And today I will present a project that is a very huge collaborative effort that is about cGAS-like receptors in Drosophila and cyclic dinucleotides. So my presentation is going to be based on this paper that is called "Two cGAS-like receptors induce antiviral immunity in Drosophila." And we at the Technical University of Munich came in quite late in the process for our final experiments for the publication. It is was driven by the group of Professor Hartmann in Aarhus in Denmark, where I'm from, and Professor Jean-Luc Imler in Strasbourg, and also Hua in China. So it is hard to stress very much that this is their project, and I was very happy to be able to assist them in their final stages of this manuscript. So actually, we already touched upon immunity half an hour ago, immunity in plants. In mammals, we have a very similar system. We call it the innate immune system. It's very conserved, which is why you can also find the same concepts in plants that we just heard. So the concept is that you have pathogens that have certain specific patterns. We call pathogen-associated molecular patterns, or PAMs for short. And they are recognized by pattern recognition receptors, which are conserved receptors that recognize these patterns. And then they start a signaling cascade when recognized, which then leads to the transcription of a lot of genes that in total gives us an antiviral state in the cell, or an antipathogenic cell state in the cell. For viruses, the most abundant PAM is viral nucleic acids, which are what we are going to talk a little bit about today. So the different types of nucleic acids have different receptors. So they are both receptors for RNA and DNA, and we are going to specifically talk about receptors for DNA today. So DNA viruses, how are they detected in the cell? In mammals, the pathway goes that the DNA is sensed by an enzyme that we call cGAS, or cGAS. It is what's short for it. Yeah, it produces. So what it does is that cGAS recognizes double-stranded DNA, and then it sensitizes cGAM, which is a cyclic dinucleotide that is special in a way that it has a mixed linkage. So it is a 2', 3' linkage, so it go like this all the way around, of this cyclic dinucleotide. And this is detected by what we call an adaptive protein that's called STING. And STING is then the one that relays the signal and in the end activates the transcription that produces the antiviral state of the cell. So our collaborators are very interested in the fruit fly, so Drosophila. And Drosophila melanogaster is known to have a homologue of STING. And from a previous publication, one of the collaborators also established that when you inject cyclic nucleotides, so cGAM, into the cells together with a virus, then you can see here that the induction of the introduction of the cyclic nucleotides protects against the infection. So you can see here is the replication of Drosophila C virus. And so when you pre-treat here with the cGAM, this is days post-infection down here. So at day 2 and 3, you can see that you have a reduction in the viral replication in the flies. And when you take out the Drosophila STING, then this protection is gone, which means that this is dependent on the protective abilities of cGAM is dependent on STING. So what our collaborators then wanted to know was, does Drosophila then have an enzyme similar to cGAS which can produce these cyclic dinucleotides, or maybe more specifically can produce cGAM? So they did an alignment of different genes. So you have the different mammalian cGAS enzymes, and then different genes from Drosophila. The cG7194 is a known homologue of cGAS, but the problem with this known homologue is that it has no enzymatic activity and no protection against viruses. But in this paper, we identified 4 other gene products that are related to cG7194 and cGAS, and especially the 2 that's called cG12970 and cG3042. 24. Sorry, that was misspelled down here. They have what's interesting about those is that they have a conserved active site that would be compatible with cyclic dinucleotide synthase activity. So those were the ones that were continued with, and for simplicity, so we don't have to say a lot of numbers, they have been called CTLR2 and CTLR1 for cGAS-like receptor 1 and 2. Very simple. So the first thing they did in France, plus work at the IMLA lab, was that they made transgenic flies that overexpress CTLR1 and CTLR2 by a heat activation system. So it means that when the flies are change to 29 degrees, then they start expressing the protein, the proteins. And what we can see here at mRNA level is that when they move to 29 degrees, you get a very nice upregulation of the two proteins, so the 1 and the 2, and also of this is a enzymatic inactive mutant that is made in parallel for control. So they're both expressed, and what we can see here is that when you overexpress the cGAS-like receptors, you get an upregulation of STING expression itself, and you also get an upregulation of STING-regulated genes, so the genes that would be induced by the activation of STING, which is very similar to how you would see for a human cGAS enzyme reaction. So the transgenic flies were then infected with viruses. So here you can see that when you take the viral replication of BSV and PCV, you get for CDLR1 and 2, you get a reduction in the viral replication for both of them, which is then abolished when you take out the enzymatic activity, the assumed enzymatic activity of the catalytic site in the enzyme. And for Drosophila C virus, it is unfortunately only significant for CDLR2. But what is very interesting here is that the survival of the flies improves markedly when they have expression of the cGAS-like receptors. So where we came in the last stages of this project was that we wanted to detect if these enzymes, so the cGAS-like receptors in Drosophila, if they actually produce versions of the cyclic dinucleotides cGAS. And the method we use is that we take human cells. I will get in the individual experiments exactly what type of cells they were. And then we lyse them in methanol, and we make an ATP gamma S spike in so that we can use that for normalization in the end after the purification. Then we do a solid phase extraction, which is a weak anion exchange sorbent, so essentially just a spin column. I concentrate by Speedvac, and then we send it to Physing, so to the BayBioMS, where the metabolomics core facility measures the nucleotides, the cGAM specifically, on a targeted approach on LC-MS. So what you see here is that when you take, so this is based on a human cell system, because that's where we made it work the best. So just don't be confused without fly. But then what you can see here is the chromatogram from when you take just cell lysates that are not expressing anything, and you do a spike in with 2 different types of scan. So we've already come across the 2', 3' scan, and then you also have the inverse one, which is the 3', 2' scan, and that becomes important in a moment. Because what we found out was that human cGAS, when overexpressed, or stimulated with DNA, actually, they only produce, and that's also what's known and very well established, produce 2', 3' cGAM. But what we saw very specifically very interestingly for CDLR2 is that it is able to, when we detect it on the mass spec, able to produce simply by overexpression both the 3' 2' gap and the 2' 3' gap, whereas the CDLR1 does not seem to produce any of them, at least in this system. I have to add that this is a human system where we introduce a Drosophila expression of Drosophila receptors, so it can be that we do not have the right components or stimulants for this receptor. As opposed to the human cGAS, it does not seem like the CDLR2 is dependent on DNA. Yeah, but what we found out afterwards is that the CDLR1 actually, so the one that did not produce anything in response to DNA or by overexpression, if we simulate with RNA, we can actually get a production of 3' 2' again, but not 2' 3' again. So it seems to be an RNA dependent production for CDLR1. So since there is no dependency on CDLR2 on DNA, and it somehow still expresses, makes cGAM by overexpression, we wanted to make sure that it is nucleic acid dependent. So we took and mutated what we think is the putative nucleic acid binding sites as compared when you compare it to human cGAS. And when that is mutated, mutations down here, we no longer get an activation of CDLR2, and this is an activation of a sting response gene. So, in conclusion, what this paper nicely figured out is that Drosophila do have sensors similar to cGAS. In fact, they have two. CDLR1 detects long double-stranded RNA and produces 3' 2' cGAM, which then activates Drosophila sting response. Whereas CDLR2 produces both the classical 2' 3' cGAM and the 3' 2' cGAM, but to a yet unknown nucleic acid. Okay. And that's where I'm gonna end today. I'm just gonna say thank you for your attention, and yes.
Okay, thank you very much for the very interesting overview of this joint collaboration.